Digestive system. Introduction. Digestion is defined as the process by which food is broken down into simple chemical substances that can be absorbed and used as nutrients by the body. In the digestive system, digestive process is accomplished by mechanical and enzymatic breakdown of food into simpler chemical compounds. A normal young healthy adult consumes about 1 kilogram of solid diet and about 1 to 2 liter of liquid diet every day. Anatomy of the digestive system. Digestive system is made up of gastrointestinal tract or alimentary canal and accessory organs. G.I.T is a tubular structure extending from the mouth up to anus. It have 30 feet length. It's open to the external environment on both ends. G.I.T is formed by two types of organs. Primary digestive organs. Accessory digestive organs. Primary digestive organs. In these organs digestion takes place actually. Mouth, pharynx, esophagus, stomach, small intestine, large intestine. Accessory digestive organs. Those which help primary digestive organs in the process of the digestion. Accessory digestive organs. Teeth, tongue, salivary glands, exocrine part of pancreas, liver, gallbladder. Wall of G.I.T. Mucus layer. Submucous layer. Muscular layer. Serous or fibrous layer. Mouth and salivary gland. Mouth is known as oral cavity or buccal cavity. It is formed by cheeks, lips, and palate. It encloses the teeth, tongue, and salivary glands. Mouth opens anteriorly to the exterior through lips and posteriorly through fauces into the pharynx. Buccal cavity. Function of mouth. Primary function of the mouth is eating. Ingestion of food materials. Chewing the food and mixing it with saliva. Appreciation of taste of the food. Transfer of food, bolus, to the esophagus by swallowing. Role in speech. Social function like expression. Salivary glands. Three major salivary glands. Parotid gland. Submaxillary or submandibular glands. Sublingual glands. Parotid gland. Parotid glands weight 20 to 30 g. And length 35 millimeters to 40 millimeters. The parotid gland opens inside the cheek against the upper second molar teeth. Viaduct of Stenson duct. Submaxillary glands. Its weight is 8 to 10 gram. And length 40 millimeters long. The ducts opens at the side of frenulum of tongue, by mean of small opening on the submit of papilla called caruncula sublingualis. Viaduct of Wharton duct. Sublingual gland. It is the smallest salivary glands. It has weight 2 to 3 gram. It's open in the mouth via 5 to 15 small ducts called ducts of Rivenus. These ducts are open on small papillae beneath the tongue. One of the ducts is larger and it is called Bartholin duct. Properties and composition of saliva. Its produces volume is 1000 ml to 1500 ml per day. And have a pH, 6.35 to 6.85. Its tonicity is hypotonic to plasma. Saliva contains 99.5% water and 0.5% solids. Two types of solids are present in the saliva: organic and inorganic. Stomach. Stomach is a hollow organ situated just below the diaphragm on the left side in the abdominal cavity. Volume of empty stomach is 50 ml. It can expand to 1 L to 1.5 L and still further capable up to 4 L. Parts of the stomach. Cardiac region. Fundus. Body or corpus. Pyloric region. Glands of stomach. Gastric glands. Glands of the stomach or gastric glands are tubular structures made up of different types of cells. These glands open into the stomach cavity via gastric pits. Classification of glands of the stomach. Gastric glands are classified into three types, on the basis of their location in the stomach. 1. Fundic glands or main gastric glands or oxyntic glands, situated in body and fundus of stomach. 2. Pyloric glands, present in the pyloric part of the Stomach. 3 cardiac glands, located in the cardiac region of the stomach. Function of stomach. Storage function. Food is stored in the stomach for a long period, i.e. for 3 to 4 hours, and emptied into the intestine slowly. The maximum capacity of stomach is up to 1.5 L. Slow emptying of stomach provides enough time for proper digestion and absorption of food substances in the small intestine. Formation of chyme. Peristaltic movements of stomach mix the bolus with gastric juice and convert it into the semi-solid material known as chyme. Gastric juice contains of 99.5% water and solids 0.5%. In solids, organic and inorganic substances. Organic substances. Exocrine part of pancreas. The exocrine function is concerned with the secretion of digestive juice called pancreatic juice. Exocrine part of pancreas resembles salivary gland in structure. It is made up of acini or alveoli. Pancreas is supplied by both sympathetic and parasympathetic fibers. Sympathetic fibers are supplied through splanchnic nerve and parasympathetic fibers are supplied through vagus nerve. Pancreatic juice. Its volume, 500 to 800 ml/day. Reaction, highly alkaline with a pH of 8 to 8.3. Specific gravity, 1.010 to 1.018. Functions of pancreatic juice. Pancreatic juice plays an important role in the digestion of proteins and lipids. It also has mild digestive action on carbohydrates. Pancreatic juice has digestive functions and neutralizing action. Major proteolytic enzymes of pancreatic juice are trypsin and chymotrypsin. Other proteolytic enzymes are carboxypeptidases, nuclease, elastase and collagenase. Neutralizing action of pancreatic juice. When acid chyme enters intestine from stomach, pancreatic juice with large quantity of bicarbonate is released into intestine. Presence of large quantity of bicarbonate ions makes the pancreatic juice highly alkaline. This alkaline pancreatic juice neutralizes acidity of chyme in the intestine. Neutralizing action is an important function of pancreatic juice because it protects the intestine from the destructive action of acid in the chyme. Mechanism of pancreatic secretion. Pancreatic enzymes are synthesized in ribosomes, which are attached to the endoplasmic reticulum of acinar cells in pancreas. The raw materials for the synthesis of pancreatic enzymes are the amino acids, which are derived from the blood. After synthesis, the enzymes are packed into different zymogen granules by Golgi apparatus and stored in cytoplasm. When stimulated, the acinar cells release zymogen granules into the pancreatic duct. From the granules, the enzymes are liberated into intestine. Secretion of bicarbonate ions. Mechanism of bicarbonate secretion. Carbon dioxide derived from blood or metabolic process combines with water inside the cell to form carbonic acid in the presence of carbonic anhydrase. Carbonic acid dissociates into hydrogen and bicarbonate ions. Bicarbonate ions are actively transported out of the cell into the lumen. Hydrogen ion is actively transported into blood in exchange for sodium ion. Sodium ion from the cell is transported into the lumen, where it combines with bicarbonate to form sodium bicarbonate. Because of the loss of sodium and bicarbonate ions from the blood, there is some disturbance in the osmotic equilibrium of the blood. To maintain the osmotic equilibrium, water leaves the blood and enters the lumen of pancreatic duct by osmosis. In the lumen, bicarbonate combines with water forming the solution of bicarbonate. Regulation of pancreatic secretion. Secretion of pancreatic juice is regulated by both nervous and hormonal factors. Stages of pancreatic secretion. Cephalic phase. Gastric phase. Intestinal phase. These three phases of pancreatic secretion correspond with the three phases of gastric secretion. Liver and gallbladder. Liver is a dual organ having both secretory and excretory functions. It is the largest gland in the body, weighing about 1.5 kilograms in man. It is located in the upper and right side of the abdominal cavity, immediately beneath diaphragm. Liver is made up of many lobes called hepatic lobes. Each lobe consists of many lobules called hepatic lobules. Hepatic lobule is the structural and functional unit of liver. There are about 50,000 to 100,000 lobules in the liver. The lobule is a honeycomb-like structure and it is made up of liver cells called hepatocytes. Biliary system. Biliary system or extrahepatic biliary apparatus is formed by gallbladder and extrahepatic bile ducts outside the liver. Right and left hepatic bile ducts which come out of liver join to form common hepatic duct. It unites with the cystic duct from gallbladder to form common bile duct. All these ducts have similar structures. Common bile duct unites with pancreatic duct to form the common hepatopancreatic duct or ampulla of Bader, which opens into the duodenum. Properties and composition of bile. Volume, 800 to 1200 ml/day. Reaction, alkaline. pH, 8 to 8.6. Specific gravity, 1.010 to 1.011. Color, golden yellow or green. Bile contains 97.6% of water and 2.4% of solids. Solids include organic and inorganic substances. Secretion of bile. Bile is secreted by hepatocytes. The initial bile secreted by hepatocytes contains large quantity of bile acids, bile pigments, cholesterol, lecithin and fatty acids. From hepatocytes, bile is released into canaliculi. From here, it passes through small ducts and hepatic ducts and reaches the common hepatic duct. From common hepatic duct, bile is diverted either directly into the intestine or into the gallbladder. Sodium, bicarbonate, and water are added to bile when it passes through the ducts. These substances are secreted by the epithelial cells of the ducts. Addition of sodium, bicarbonate, and water increases the total quantity of bile. Storage of bile. Most of the bile from liver enters the gallbladder, where it is stored. It is released from gallbladder into the intestine whenever it is required. When bile is stored in gallbladder, it undergoes many changes both in quality and quantity. Such as, volume is decreased because of absorption of a large amount of water and electrolytes, except calcium and potassium. Concentration of bile salts, bile pigments, cholesterol, fatty acids and lecithin is increased because of absorption of water and electrolytes. The pH is decreased slightly. Specific gravity is increased. Mucin is added to bile. Function of the bile salts. Emulsification of fats. Absorption of fats. Choleretic action. Cholagogue action. Laxative action. Prevention of gallstone formation. Small intestine. Small intestine is the part of gastrointestinal, GI, tract, extending between the pyloric sphincter of stomach and ileocecal valve, which opens into large intestine. It is called small intestine because of its small diameter, compared to that of the large intestine. But it is longer than large intestine. Its length is about 6 meters. Important function of small intestine is absorption. Maximum absorption of digested food products takes place in small intestine. Small intestine consists of 3 portions. Proximal part known as duodenum. Middle part known as jejunum. Distal part known as ileum. Intestinal villi and glands of small intestine. Mucous membrane of small intestine is covered by minute projections called villi. The height of villi is about 1 millimeter and the diameter is less than 1 millimeter. Villi are lined by columnar cells, which are called enterocytes. Each enterocyte gives rise to hair-like projections called microvilli. Villi and microvilli increase the surface area of mucous membrane by many folds. Within each villus, there is a central channel called lacteal, which opens into lymphatic vessels. It contains blood vessels also. Types of cells interposed between columnar cells of intestinal glands. Argentophane cells or enterochromophane cells, which secrete intrinsic factor of Castle. Goblet cells, which secrete mucus. Pannate cells, which secrete the cytokines called defensins. Properties and composition of succus entericus. Secretion from small intestine is called succus entericus. Volume, 1800 ml/day. Reaction, alkaline. pH, 8.3. Succus entericus contains water, 99.5%, and solids, 0.5%. Solids include organic and inorganic substances. Bicarbonate concentration is slightly high in succus entericus. Enzymes of succus entericus act on the partially digested food and convert them into final digestive products. Enzymes are produced and released into succus entericus by enterocytes of the villi. Proteolytic enzymes present in succus entericus are the peptidases, which are given in. These peptidases convert peptides into amino acids. Lactase, sucrase, and maltase convert the disaccharides, lactose, sucrose, and maltose, into two molecules of monosaccharides. Dextrinase converts dextrin, maltose, and maltriose into glucose. Trihalase or trihalose glucohydrolase causes hydrolysis of trihalose, carbohydrate present in mushrooms and yeast, and converts it into glucose. Large intestine. Large intestine or colon extends from ileocecal valve up to anus. Large intestine is made up of the following parts. Cecum with appendix. Ascending colon. Transverse colon. Descending colon. Sigmoid colon or pelvic colon. Rectum. Anal canal. Structure of wall of large intestine. Wall of large intestine is formed by 4 layers of structures like any other part of the gut. Serous layer. It is formed by peritoneum. Muscular layer. Smooth muscles of large intestine are distributed in 2 layers, namely the outer longitudinal layer and inner circular layer. The longitudinal muscle fibers of large intestine are arranged in the form of 3 long bands called taenia coli. The length of the taenia coli is less when compared to the length of large intestine. Because of this, the large intestine is made into series of pouches called haustra. Submucous layer. It is not well developed in large intestine. Mucus layer. The crypts of Lieberkühn are present in mucosa of large intestine. But the villi, which are present in mucous membrane of small intestine, are absent in the large intestine. Only mucus-secreting glands are present in the mucosa of large intestine. Secretion of large intestine. Large intestinal juice is a watery fluid with pH of 8.0. Composition of large intestinal juice. Large intestinal juice contains 99.5% of water and 0.5% of solids. Digestive enzymes are absent and concentration of bicarbonate is high in large intestinal juice. Functions of large intestinal juice. Neutralization of acids. Strong acids formed by bacterial action in large intestine are neutralized by the alkaline nature of large intestinal juice. The alkalinity of this juice is mainly due to the presence of large quantity of bicarbonate. Lubrication activity. Mucin present in the secretion of large intestine lubricates the mucosa of large intestine and the bowel contents, so that the movement of bowel is facilitated. G.I.T hormones. Gastrointestinal, G.I. hormones are the hormones secreted in GI tract. These hormones are polypeptides in nature and belong to the family of local hormones. Major function of these hormones is to regulate the secretory activities and motility of the GI tract. Cells secreting the hormones. Enteroendocrine cells. Neuroendocrine cells or APUD cells. Enterochromophane cells. Digestion, absorption, and metabolism of carbohydrates. Human diet contains 3 types of carbohydrates. Polysaccharides. Large polysaccharides are glycogen, amylose and amylopectin, which are in the form of starch, glucose polymers. Glycogen is available in non-vegetarian diet. Amylose and amylopectin are available in vegetarian diet because of their plant origin. Disaccharides. Two types of disaccharides are available in the diet. Isucrose, glucose plus fructose, which is called table sugar or cane sugar too. Lactose, glucose plus galactose, which is the sugar available in milk. Monosaccharides. Monosaccharides consumed in human diet are mostly glucose and fructose. Other carbohydrates in the diet include alcohol, 2 lactic acid, 3 pyruvic acid, 4 pectins, v dextrins, 6 carbohydrates in meat. Diet also contains large amount of cellulose, which cannot be digested in the human GI tract, so it is not considered as a food for human beings. Digestion of carbohydrate. In the mouth. Enzymes involved in the digestion of carbohydrates are known as amylolytic enzymes. The only amylolytic enzyme present in saliva is the salivary amylase or ptyalin. In the stomach. Gastric juice contains a weak amylase, which plays a minor role in digestion of carbohydrates. In the intestine. Amylolytic enzymes present in the small intestine are derived from pancreatic juice and succus entericus. Carbohydrates are absorbed from the small intestine mainly as monosaccharides, viz. glucose, galactose, and fructose. Absorption of glucose. Glucose is transported from the lumen of small intestine into the epithelial cells in the mucous membrane of small intestine, by means of sodium cotransport. Energy for this is obtained by the binding process of sodium ion and glucose molecule to carrier protein. From the epithelial cell, glucose is absorbed into the portal vein by facilitated diffusion. Sodium ion moves laterally into the intercellular space. From here, it is transported into blood by active transport, utilizing the energy liberated by breakdown of ATP. Absorption of galactose. Galactose is also absorbed from the small intestine in the same mechanism as that of glucose. Absorption of fructose. Fructose is absorbed into blood by means of facilitated diffusion. Some molecules of fructose are converted into glucose. Metabolism of carbohydrates. Metabolism is the process in which food substances undergo chemical and energy transformation. After digestion and absorption, food substances must be utilized by the body. The utilization occurs mainly by oxidative process in which the carbohydrates, proteins, and lipids are burned slowly to release energy. This process is known as catabolism. Part of the released energy is utilized by tissues for physiological actions, and rest of the energy is stored as rich energy phosphate bonds and in the form of proteins, carbohydrates, and lipids in the tissues. This process is called anabolism. Digestion, absorption, and metabolism of proteins. Enzymes responsible for the digestion of proteins are called proteolytic enzymes. In the mouth. Digestion of proteins does not occur in mouth, since saliva does not contain any proteolytic enzymes. So, the digestion of proteins starts only in stomach. In the stomach. Pepsin is the only proteolytic enzyme in gastric juice. Renin is also present in gastric juice, but it is absent in human. In the small intestine, most of the proteins are digested in the duodenum and jejunum by the proteolytic enzymes of the pancreatic juice and succus entericus. Proteolytic enzymes in pancreatic juice. Pancreatic juice contains trypsin, chymotrypsin, and carboxypeptidases. Trypsin and chymotrypsin are called endopeptidases, as these two enzymes break the interior bonds of the protein molecules. Proteolytic enzymes in succus entericus. Final digestion of proteins is by the proteolytic enzymes present in the succus entericus. It contains dipeptidases, tripeptidases, and aminopeptidases. Absorption of proteins. Proteins are absorbed in the form of amino acids from small intestine. The levo amino acids are actively absorbed by means of sodium cotransport, whereas the dextro amino acids are absorbed by means of facilitated diffusion. Absorption of amino acids is faster in duodenum and jejunum and slower in ileum. Digestion, absorption, and metabolism of lipids. Lipids are mostly consumed in the form of neutral fats, which are also known as triglycerides. Triglycerides are made up of glycerol nucleus and free fatty acids. Triglycerides form the major constituent in foods of animal origin and much less in foods of plant origin. Apart from triglycerides, usual diet also contains small quantities of cholesterol and cholesterol esters. Dietary fats are classified into two types, 1. Saturated fats, 2. Unsaturated fats. Saturated fats. Saturated fats are the fats which contain triglycerides formed from only saturated fatty acids. The fatty acids having maximum amount of hydrogen ions without any double bonds between carbon atoms are called saturated fatty acids. Unsaturated fats. Fats containing unsaturated fatty acids are known as unsaturated fats. Unsaturated fatty acids are fatty acids formed by dehydrogenation of saturated fatty acids. Unsaturated fats are classified into three types, 1. Monounsaturated fats, 2. Polyunsaturated fats, 3. Trans fats. Monounsaturated fats. Unsaturated fats which contain one double bond between the carbon atoms are called monounsaturated fats. Polyunsaturated fats. Unsaturated fats with more than one double bond between the carbon atoms are called polyunsaturated fats. Polyunsaturated fats belong to the family of essential fatty acids required in diet. Polyunsaturated fats are of two types. Omega-3 fats or omega-3 fatty acids having double bond in the third space from the end of the carbon chain. Omega-6 fats or omega-6 fatty acids having double bond in the sixth space from the end of the carbon chain. Both omega-3 and omega-6 fatty acids are beneficial to the body. However, consuming too much of omega-6 fatty acids results in hazards than benefits. So, the diet containing 3:1 ratio of omega-6 to omega-3 fatty acids is often recommended by experts. Trans fats. Trans fats or trans fatty acids are unsaturated fatty acids, with molecules containing trans, across or opposite side, double bonds between carbon atoms.